Muy buenas tardes a todos. Muchas gracias por estar presentes en nuestro seminario. Hoy tenemos un seminario de lujo porque el doctor Rupesh Kariyat nos acompaña hoy, además, tiene dos bonitas situaciones. La primera es que Rupesh es mi colega, es nuestro colega en un gran proyecto de ecología química de las chinches que transmiten Chagas. Hemos venido trabajando súper lindo con él a pesar de la pandemia. Se los recomiendo mucho como colega, como colaborador. Es muy entusiasta y es muy buena gente. Y eso me lleva al otro punto. Es una gran persona. Entonces, estamos de plácemes hoy con su presencia. Rupesh Kariyat, del doctor Kariyat, es profesor asistente o asociado de biología en la Universidad de Texas en el campus del Valle de Río Grande. Ahí ya tiene algunos años, pero su origen es hindú. Él hizo su licenciatura en la India y de ahí se movió a hacer sus posgrados y una estancia postdoctoral en la Pennsylvania State University y también en la Swiss Federal Institute of Technology en Suiza. Tiene varias publicaciones en el campo de lo que vamos a escuchar, que es la de ecología química en las interacciones entre polinizadores, plantas, etcétera. Ahí tiene un vínculo muy interesante con las áreas aplicadas, entonces sí está muy lindo pues lo que él hace. Y ha accedido muy amablemente a presentarnos esto en este ciclo de seminarios que tenemos sobre la ecología de insectos en ambientes cambiantes. Entonces, sin más, les voy a dejar con el doctor Kariyat. Como siempre, pueden usar el chat en la pantalla para plantearnos sus comentarios, sus preguntas, las cuales leeré al final. O también pueden tomar directamente el micrófono y plantearle los cuestionamientos a Rupesh. Entonces, ya con eso les dejo. Rupesh, thank you very much again for being here, and it's all yours. Please go ahead.
Thank you, Alex, for the invitation. I'm still getting used to it. Had been like a year and a half giving talks through Zoom. It's a little bit hard without actually seeing the people, but I'm going to give my best try here. So I'm going to talk about two things. One is a central theme of my research, which started when I was a PhD student at Penn State. And then what I derived from that line of research and how once I got the independent position, how I have modified or found offshoots from that theme of research and what we are doing as a lab now in Texas. So the title is Using Chemical Ecology and Field Biology to Understand the Role of both Herbivores and Pollinators in a plant mating system evolution. So it's heavily focused on the plant side, but using the two selection pressures, the herbivores and pollinators. We don't really have a lot of time to discuss about pollinators, so most of the work I'm going to show is about herbivores. So there are a few interests that I had been working on since grad school. And again, due to time constraints, we are going to focus more on the first part, which is inbreeding and herbivore-pollinator interactions. I'm also interested in looking at extrafloral nectaries that the plants produce and how does multi-trophic interactions get affected by this production of extrafloral nectaries, the content, the predators, all those things. Herbivore-pollinator interactions and flower color polymorphism. So since the time of Darwin, we have seen this, how pollinators are being beguiled by differences in floral colors and how do they respond to that. So that's another area that I'm interested in. So cutting back or going back to the most interesting or the most important thing that we are going to spend most of our time today is about inbreeding and inbreeding specifically in plants. So we all know that plants display genetic variation in traits like any other organisms, and that can also influence the performance of herbivores. This is something that people have started to look at from early 90s. Then there was a seminal paper in 2002 from Mickey Eubanks and David Carr. They set this stage for exploring this at more ecological level. So we see this variation at both population and individuals. Inbreeding is a common example for this genetic variation. So 25% of all flowering plants, what is that, 250,000 plants, they actually undergo some sort of selfing. So some sort of inbreeding. It could be selfing, but it could also be mating within related, genetically related individuals, especially in founding populations where you start with only a few number of alleles. If they don't inbreed, then they can go locally extinct. It's very important for invasive weeds or in general any weed species. Unlike animals, which also tend to inbreed or we can force to inbreed them, plants not necessarily have a phenotype for inbreeding. The plants may be smaller, they may be shorter, they may not produce that many fruits or flowers, but sometimes we don't really have a phenotype like to show this. So for example, this is an inbred tiger. The name of the tiger was Kenny. It was the Kenny was inbred forcefully, and you can see that the facial structure, the bone structure of Kenny is quite deformed. It's very clear that Kenny suffered from some deformities, and possibly due to the deleterious alleles which were masked if it was heterozygous, but because it's more homozygous, these deleterious alleles come up and then they cause these defects. Kenny died. It was sterile, of course. Kenny died at a very young age. So if you take a step back and think that Kenny was an example of an inbred plant, then we could start asking questions from a genotype to the actual realized fitness in the form of seeds or the germination of progeny. How can inbreeding affect these traits? And if we, since I'm an insect plant ecologist, I always want to look at from the perspective of insects. So if we look at herbivores, which are causing damage and sort of exerting pressure, and on the same way, pollinators, which are actually helpful, and then they help the plants to get fertilized and produce progenies. So we can ask questions once we have inbred plants and outbred plants, then we can start to use these herbivores and pollinators and then ask these very detailed questions on ecological interactions that they mediate with both inbred and outbred plants. So I did my PhD under Dr. Andy Stevenson and Professors Mark Meshker and Consuelo de Moraes at Pennsylvania State University. And Andy had been working on inbreeding for a while, but mostly from a plant reproductive biology perspective, not necessarily the ecological interactions. There was one student who worked on this before me, and then I started on this. So this was my study system. The insect is still our most important study system. The joke in my lab now is, regardless of what we do, we have to work with the Manduca. So horse nettle, which is an herbaceous perennial, it's also invasive. It's noxious in many parts of the world. Not so bad like another plant that I'm going to discuss later in the talk. And the caterpillar or the insect that I used was Manduca sexta, tobacco hornworm, which is something that you guys are also very familiar. It's something that eats your Jimson weed datura and also a lot of tomatoes. It's called tobacco hornworm. They are both native to United States and Mexico. It can cause severe damage. It's estimated that a single Manduca caterpillar, if it is fourth or fifth stage, can eat a fully grown tomato plant in 24 hours. So it's pretty bad. So like any other incoming PhD student, the first question to ask was, how bad is inbreeding using a field experiment, since we are ecologists. So I did this 2-year field experiment, and then I established the fact that inbreeding is pretty bad. So we looked at fitness measures, and we also looked at, if we exclude herbivory, how does that affect inbreeding? So we saw that it enhances inbreeding effects. And then in general, inbred plants in the field suffered higher levels of herbivory. And outbred plants, when you exclude herbivory, they were actually producing a lot more fruits and seeds. The follow-up question was, how bad is this if we ask a specific question on, are inbred plants more susceptible? So we started looking for evidence by looking at damage levels on inbred and outbred plants. So we would set these plants outside, we would grow them, and we would continuously collect data on what are the different types of insects that feed on them, but also what is the scale of that damage. And then we were able to correlate that with the gene expression by doing like a massive microarray. So we did a transcriptome of horse nettle. Nobody produces the transcriptome chip for horse nettle, but we could hybridize with the tomato chip. And then we found that, yes, inbred plants, they are more susceptible and the outdoor plants are much more resistant. But if you look at the gene expression, you can see that constitutively the genes which are involved in defense pathways are much more upregulated in outdoor plants. So they are inherently better at defending against herbivory. So now that we have established that, yes, inbreeding is bad, inbreeding affects fitness, but also inbreeding has a disproportionate effect on the susceptibility of herbivores on these plants. So the lab that I worked was also a chemical ecology lab, and one of the main focus of chemical ecology labs is look at volatiles. So these volatiles are compounds that are emitted by plants as a part of their regular metabolism. They are secondary metabolites. They are also defense compounds. They can affect, or they can function as compounds that would attract predators of herbivores which are feeding them, but also selectively repel some of the herbivores, but also attract pollinators. We all know about volatile compounds. When we take a cilantro leaf and a tomato leaf, and then we squish them in our hand, they smell different. Even without squishing, they also smell different. So when we squish them, we are breaking up membrane lipids, and then that releases a lot more compounds than they would have when they were undamaged. So volatiles have been since so the best paper, or the cornerstone paper, came out in 1993 from Parr and Tomlinson. Then 1997. Then my advisors published in 1998, 2001. Then these papers set the stage, showing that the volatile signature of plants are very dynamic. So it can be altered by abiotic stress, biotic stress. Within biotic stress, if an aphid feeds on a plant, they would emit a different set of compounds. And if caterpillars feed on plants, they would emit a different set of compounds. And within caterpillars, two different types of caterpillars can emit different types of compounds. So we had all these excellent papers which came out before. So the obvious question was to ask, does inbreeding affect the inherent ability of plants to emit these volatile signatures or volatile signaling molecules, right? So we started looking at that. This is the volatile collection setup. So I worked with Alex on one of these projects with context, and my job is actually to look at volatile compounds. So this was the setup in my previous lab. My setup here is not as big as this. We don't have a separate room just for the volatiles. So we push clean air, and then we pull, we cause a positive air pressure, and then we would suck air out of these jars, and inside the jars we set up the plants or insects or animals or whatever we want. We would change based on the size of the organism that we are keeping it inside. And while we are pulling air, we would stick a filter inside this tubing, and then this filter will trap all these volatile compounds, and then we would elute the filter using an organic solvent. We usually use dichloromethane, and then we run it on a GC with an FID. So, and then we have a mass spec that would give us the compound identity. And because we had internal standards, a known amount, and with the retention time, you can quantify. So we get two things. We know what are the compounds emitted. We also know what is the relative amount of those compounds emitted. So we can ask this interesting question. If you have like 10 of these or 12 of these at the same time, we can have a replicated experiment where we have plants without any insect damage and plants with insect damage, plants from different lines. So inbred, outbred, damaged, undamaged, so all these things, right? So it's a very good tool to have. So we did that, and then we found very interesting results. So our data, or the data so far, suggests that inbreeding is just bad. So anything that is related, any trait that we are interested in, if we look at the plants, then it will be diminished or disrupted in inbred plants. But that's not entirely true. So when the plants are undamaged, so there is no caterpillars feeding on them, the inbreds actually smell more, which was weird. We didn't expect that. And then when you put caterpillars on them, both of them smell much more than when they were undamaged. That's fine, because they are inducing more compounds. It's like their 911 call to attract predators. But that increase, or that index of increase, or that induction is much lower in inbred plants. In other words, what happens is when there is no damage, when the the outbred plants are quiet. They are not smelly. And when inbred plants are smelly or leaky, the volatile disruption is leaky, then they actually attract more herbivores. And when it is time for them to make their 911 call because they are being eaten by this herbivore, they are trying, but they are not loud enough. And at that time, the outbred plants are much louder. So when they have to be quiet so that they don't have to be they won't get eaten by someone, they are super quiet. But when it is time to ring that bell or call 911, they are like super loud. So it completely works backward for inbred plants in volatile emission. So the obvious question was, does it actually matter for the caterpillars? So we did these experiments where we would set up like 200 caterpillars, one caterpillar at one time, and do choice assays. So this was like, it was just one figure, but it took like a year to do it because you have all these treatments from different maternal lines of inbred and outbred plants, and then you would have one inbred plant and one outbred plant, and that is whole plant, and you would release a caterpillar and then see where it goes. Then you would cut leaf discs from them and then put them in a petri dish to see if you can even replicate that with actual punched holes and see. So it was super consistent. There was no question. So if you are a leaky and if you are inbred, then they go to you. But if you are damaged by a caterpillar, then they produce much more volatiles. And I will show you the significance of that in a minute in a field experiment. But here is a short video where you can actually see. So inbred is S and outbred is X. So this caterpillar, this caterpillar is second instar caterpillar. It has never seen a plant before. It was always been on artificial diet, and that's an inbred plant and that's an outbred plant. So, and we just release this caterpillar here.
Did you ever have to make up your mind? Pick up that one and leave the other behind. It's not often easy, and not often kind. Did you ever have to make up your mind? Did you ever have to finally decide and say yes to one and let the other one ride? There's so many changes, tears you must hide. Did you ever have to finally decide? Sometimes there's one ridiculous cute as a berry, with hair down to here and plenty of money. And just when you think she's that boy in the world, your heart gets stolen by some massive little girl. And you know you better make up your mind and pick up a fork and leave the other behind. It's not often easy and not often kind. Did you ever have to make up your mind?
All right, so two things. One, my advisor made me promise that every time I show this video, I should say that the credit for the song goes to him. So he found the song. The video is mine, but the song is his. So that's the first thing. The second thing is, there are two interesting things here. One is that you can see that the caterpillar is actually lifting its head, and then it is doing this like semicircular motion and it's looking for volatile cues. So the cues are local, but also it comes as volatile puffs, right? It comes and hits you. And the second thing is they just don't go in a straight line. It went to the center of the petri dish, then it made the decision to where it would like to go. So the volatile cues, while they are local, but they are mixing with the air, there is a plume going, but they are so distinct and different from each other. And the caterpillar is able to use, it's a little bit hard for the caterpillar in the sense that it has never encountered a plant. So this is hardwired, and it has to lift its head and it has to search for these cues, and then it finally goes to the inbred plant. So in the field, we see that the inbred plants attract more herbivores. In the lab, we can replicate that with a completely uninformed caterpillar. And then we want to see whether this is just for the herbivores. So far, we have only looked at this caterpillar. But in the field, it's community dynamics of different insects. So we have predators, we have parasitoids, we have herbivores, we have everything. So we set up these traps, and then we would collect. So we have inbred plants, outbred plants, inbred damaged for 2 days with the Manduka, outbred damaged with 2 days with the Manduka, and would set them up. And it's in a squash field, so you have, there is some activity, but it is far enough that the volatile plume doesn't mix too much and it's still local. And then we started looking at what are the insects that we would get it. So it's a busy slide, but the point here is in inbred plants, when you have damage, even if they have damage or if they are undamaged, there is this enhanced attraction of herbivores, which is bad for them because they are leaky. And when it is time to attract natural enemies that would come and parasitize the caterpillar or eat the caterpillar, then they don't have that power, that the induction power is much less. Outdoor plants, they are quiet, so they are less apparent to herbivores when undamaged, but when they are induced, then they are much louder and they would attract more natural enemies. So we see that with all these natural enemies, including predators and parasitoids in the 2-year field experiment. So Cortesia is one of these parasitoids that we saw in Braconidae. So now we have established that yes, in the field this is bad, in the lab this is bad, but all these experiments are done during the day. So this is all daytime experiments. I would go and close these cages with big trash cans and put like rocks on them, because in Pennsylvania during summer it's so windy in the field and the trash cans would fly away. Still remember that. So the follow-up question is, when these caterpillars become adults, these become moths. So you can see the moth here, and the moth lays eggs only at night. So it's a nocturnal moth, exclusively nocturnal moth, and it lays eggs. So if caterpillars are hardwired to identify inbred plants, which are possibly more nutritious and less defended, we all know in ecology there is this concept like the mother knows best. It has been successfully tested in different systems. So obvious question to ask was, what happens at night? So we looked at volatile compounds emitted just at night. So this is just exclusive nighttime. So there is no photosynthesis. And then what compounds are emitted. So we found that the outcrossed plants, again, when they were undamaged, they emit less, but when at night, when they are damaged, they emit much more volatiles. And what happens is this has effects on oviposition of the moths. So what happens is the moths almost exclusively dump their eggs on inbred plants, regardless of the damage. So, and they spend much more time also on inbred plants. So this experiment was done over a year, where the experiment would start at night, and then I would stay in this dark room. If it is completely dark, they won't fly well. So we have to put like a small moonlight, and then I would sit near this cage because I had to time them. So I had to sit there and time how many seconds each moth spends, and I can only do one cage at a time. Then I have to wash it and clean it and set up another cage, and then set up new moths, new plants. So this was quite time consuming. But it was an interesting experiment. It was very clear that you would see these moths flying. So this paper, it has a video where you can see this moth would fly and then hang out on inbred plants, dump like 2 or 3 eggs, then fly out, go to an outbred plant and dump like 1 egg because you are given a choice. So on the opposite corners, diagonally opposite, you have 2 inbred plants and 2 outbred plants. We also did experiments where we only put inbred or outbred plants, not giving them a choice. So if you don't give them a choice, then they don't care. They would just dump all their eggs on inbred or outbred. Only when you are given a choice and you know that the volatile signature is different, then they actually lay their eggs on inbred plants. So the caterpillars like inbred plants, you see more damage on them. The mother likes inbred plants. The mother is dumping all its eggs on inbred plants, which obviously says that there is something about inbred plants that is very good. So why, although outbred plants are producing more volatiles, they are not laying eggs on them? Well, because it's almost like aposematism, where it's the warning signals. The volatiles that are produced by outbred plants at night are quite different, and they are sort of like herbivore repellent volatiles. So the idea is that you are giving a warning signal saying that I'm no good as food. So it's better if you have a choice to dump your eggs, the most important prized possession, on a plant where the caterpillars are more likely to succeed. So if you think about this, now we have this good evidence both from lab and field using different types of assays, which shows that it is better for the caterpillars or the herbivores to lay eggs, feed, and hang out more on inbred plants, which clearly says if this is an honest signal, then it should actually have some fitness benefits. So maybe better growth, maybe better development, and maybe better dispersal ability. So if they can colonize inbred plants more and they can eat well, they can eat more, they can become bigger, then it might be a better, for them for their survival. So that's exactly what we did. So we tested that by growing these caterpillars exclusively on inbred or outbred plants. So we put them and then we continuously collected mass and then all these other growth traits, and we found exactly the same. So the ones on inbred plants are doing extremely well. They are bigger, fatter, they pupate fast, etc. But then if you want to look at dispersal, we actually want to tag them and then let them fly, right? And then we want to track how far they would go, which is a little bit difficult. So what we did was we did respirometry. So we would fly them out. So this, you can see this jar. So we would fly them out. We would remove all the carbon dioxide from it. And we would fly the moth inside the jar for 5 minutes and look at its peak CO2 output. So peak CO2 output and total CO2 output are two indices which is indirectly related to the total flight muscle output. So if you have better flight muscles, then you can have much higher CO2. So you can see in these two graphs, the blue lines on the first one, you can see them significantly higher than the red lines which are outbred. And on the second graph, you can again see the same thing with the total carbon dioxide. So it's called a respirometry. And then there is a gene called troponin T. It has different splice forms, and these splice forms, the ratio of these splice forms also tell you whether you have better flight muscles. And we were able to show that with the fragment analysis that these flight forms, these isoforms also are in line with inbred plants are better for growth and development and dispersal. So to conclude, inbred plants have a disrupted indirect defenses. These are all indirect defenses providing better fitness for the herbivores. They have disrupted volatile emission, which is an honest signal saying inferior defenses, so the mother knows to dump all the eggs. Well, there are other defenses for the plants even before they get to this, right? So before they start feeding, they have to deal with these trichomes and spines. And so the obvious question was to ask them, like, so we damage the plants by mechanical wounding, but also we put caterpillars on them, and then we asked what happens with the trichomes. And we saw that, yes, the trichomes, the outdoor plants have more trichomes to start with, but when you induce them, they actually induce much more. Very similar to volatiles, but constitutively also they have more trichomes. So their surface defenses are very good. And what is the consequence of that? If you put caterpillars and if you start looking at when do they start feeding, you see that they start feeding much faster when there is less trichomes. So less trichomes, it's easy to get access to the epidermis and then start feeding. So this is an early instar caterpillar. So you can see how it is struggling on this plant with a lot of trichomes, right? So this early instar caterpillar clearly, and you know that it's white in color, it will turn to camouflage green only once it ingests a mesophyll. So it has to deal with this carpet of nails and then get access to the epidermis so that it can feed, and then it will change its color so that it doesn't get eaten by predators. The longer it takes, it's much difficult. It could get eaten, it could desiccate, and all these things. So that's a baby caterpillar. That caterpillar turns into this beast in like 12 days. So what about them? It's the same plant, but what happens is it doesn't matter. So these trichomes are there, but it doesn't matter to it. It's actually ingesting the trichomes. It's eating the trichomes. So if you think about it, so the plant defense in the form of trichomes are only useful if it is a baby caterpillar. That doesn't seem to be a good system, because if the caterpillars are somehow able to deal with it, maybe cut them off, chew them off, mow them off, then that defense is useless. Well, that's not exactly the case, because this is the inner gut, the gut lining of the caterpillar. It's called peritrophic matrix. So peritrophic matrix holds the food bolus, and it makes sure that the food doesn't get mixed with your blood or hemolymph in this case. So if you feed artificial food without any plant material to the caterpillar, this is how the electron microscopy imagery of the peritrophic matrix looks like. It's nice scaffolding. There There's nothing that looks like it's damaged or anything. And this is how it looks like when you feed them a diet with the trichomes embedded. So all these holes and these nails sticking out, they are trichomes. So what's happening is this peritrophic matrix is being ruptured or leaked, and the food is going to mix with your blood, and that leads into sepsis, immune response, reduction in growth, all these things. This is another shot of insects. So all these things, so the trichomes cannot be digested. So not only they have to deal with this leaky gut syndrome in this, but they also have to poop them out. And there is, I'm guessing there is no pleasure in pooping out nails, which goes through your intestine without getting deformed or digested. So they have to deal with that. So that's with all the herbivores. And what about the pollinators? So obvious question was to ask, like, what happens with pollinator traits? And we saw the very similar stuff, right? So the outbred flowers, so we collected volatiles from an individual flower. Unlike other flowers, this flower does not produce any nectar, and the pollen is hidden inside the poricidal anthers. So you really have to have bus pollinators to come and extract this pollen. So we found similar things, and it's again an honest signal where the bumblebees, which are the primary pollinators, they would visit more outbred flowers, they would spend more time on outbred flowers, and they would pollinate more outbred flowers. It's the complete opposite of the mother moths, right? So they would spend more time on inbred plants, they would lay more eggs on inbred leaves. In flowers, when it flowers, like the smell is, the one thing to understand is the smell is not that much. They don't smell much, unlike rose or like jasmine or lavender or something. They're not that smelly flowers, but that's enough for these bees to identify the difference. So the model is that these are all JA mediated. Jasmonic acid is a primary signaling molecule when caterpillars feed on them, and we did some network analysis of these pathways. And then this is what we find is that inbred plants lack a consistent coordinate gene expression response to herbivory. And these ramifications extend to the community, including arthropod community dynamics in the field, dispersal of the plants, and it differentially affects both pollinators and herbivores. And within herbivores, it affects the caterpillars or their young ones and these adults differentially. So having these inbred populations, which is a function of invasiveness to some extent, like Baker's law, if you think about that, then having these founding populations where you have to have these inbred plants, if you don't, you go locally extinct, will help them, the herbivores also, to establish and survive and disperse. So that's most of my PhD, some part of my postdoc work. But along that, I picked up some of these interesting things when I got this independent position. So I wanted to explore more while keeping that inbreeding core intact. I still continue to work on that. So we work on a bunch of stuff because my workforce is master's students and undergraduate students, excellent students. So almost all the stuff that I'm going to show are exclusively done by them. I just stand here and take credit for most of it. So we look at, again, plant defense and herbivore counter defense. Insect chemical ecology is where Alex and I have a project together. We don't have much, I don't have much to talk about that, so hopefully later. We look at plant secondary metabolites. We also have slowly moved into biomechanics of pollination syndromes, and then use all this in IPM, integrated pest management and biocontrol. I didn't really want to get into that, but when I got the job, that was one of the things. We didn't have someone who would do that, so I was sort of roped into these things. And then I started to enjoy it, and now I'm a big fan of those sustainable agriculture and all these things. I have a master's in agronomy, so I was able to pick up some of the science relatively easily. So we look at herbivory and we look at pollination. That's a two-word easy way to say it. So we use a bunch of different insects now. Our questions, whatever insect systems that we need, or if our question is based on a specific insect, we use it. So we are not bound by a single insect or a single study system. We ask a different, so we are more question oriented than system oriented. Whatever insect you want for answering the questions, we find them. Early and late signaling events, that's one of the things. So the fundamental question is, how do plants mount an efficient herbivore defense? We talked about trichomes, we talked about gene expression, we talked about volatiles. So what is this layering? How does this happen? What is the role of these individual layers? And how does that vary across different species within a genus and among different families, these kind of questions. And then how can we manipulate these for herbivore management? Right, again, going back to the trichomes, so we started looking at this very interesting study system. I think these plants are actually native to Mexico. These are cucurbids, Lagenaria caesarearia, and we bought an electron microscope for the lab, so we could actually do a lot of these things in-house without waiting for the core facility. So we started to look at this. So this is a graduate student, Ishween Kaur. She started looking at this morphology of these trichomes and started to find very interesting things. So this is how a caterpillar looks like on a leaf surface. So you can see that it's not a red carpet for them to walk and feed. It's pretty bad. So we started looking at these roles that they play. We had published a paper before where we show that the glandular and non-glandular trichomes. So you can see the glandular trichomes have a gland at the top and they have toxins or they have signaling molecules. The non-glandular ones, like I showed here in these both figures, they are just pointy things that would tear your mouth and your gut and then cause pain when you're trying to poop them out. So Christina Raya was another student who was an undergrad in the lab. She is now a PhD student in cell biology program at UT Austin. She and I, we started working on this and we collected these, we stripped these plants out of these trichomes using a couple of methods, and then we would add these trichomes into diet. And then we started looking at, if you look at the whole growth of the caterpillar, where is these effects, when do they start and how do they end? And we found interesting results that the glandular trichomes are not really useful as a defense if you're adding that into your diet, whereas non-glandular trichomes, which are less studied, to be honest, they are the more damaging ones. So it was sort of a confirmation to some extent using an empirical data set that these non-glandular trichomes can work in different ways by causing physical damage, causing damage inside the gut, and also inside the gut when they're trying to poop them out. So they get tore apart, their innards get torn apart by this. So these are two students who worked on this. Jesslyn wrote a book chapter on it. We were invited by Juan Nunez from Mexico to write a chapter for this. And then Sakshi came into almost a year and a half ago, and Sakshi wanted to look at these trichomes at a much broader context. She wanted to look at within a family, for example, Solanaceae family, what is the diversity of these trichomes, and does it matter like the density of the trichomes on the different sides of the plant, like abaxial and adaxial side? And Ishween continued to work on this thing. So Sakshi's first paper came out, and where she shows that so the plants, especially in the Solanaceae family, they have very different density of trichomes on both the abaxial, which is the underside, and the adaxial, which is the above side. And you always see caterpillars hanging out on the abaxial underside. Well, it has been speculated that it is to avoid a lot of heat, which is true, and also to avoid predators, which is another point why they are hanging out. So we asked the question, okay, so first we had been documenting these caterpillars, and we found that like 90% of them are always seen on the underside. But then we asked the question like, so what happens if you're forced to feed on the underside? So we did these taping experiments where they only had access to one side, and then she put the caterpillars and saw how they grew and all. And consistently she found that hanging out on the underside comes at a cost. If you were only feeding on the underside, you grow slowly when compared to if you were feeding on the above side. You can also see the difference in trichome density. So a hardwired choice by the caterpillars by feeding on the underside comes at a cost by a delay in growth and development. So once they have completed the first step, which is the first hurdle, which is the trichomes, then they start to bite. So what happens at the point of biting? And Gansha Gandhi, who is now doing PhD at Max Planck in Germany, Dr. Sahu's student, so we collaborated with them, and we gave them manduga, this caterpillar saliva or oral secretions, and we wanted them to, so they have this very nice techniques in cell biology where this is their expertise. They look at reactive oxygen species generation, calcium ion flux, things like that, which are primary and secondary messengers in plant defenses, right? So we gave them, and they had these different types of caterpillars. We gave them oral secretion from caterpillars which were fed on diet without any plant material, and caterpillars which were fed on plants. And then when we gave the saliva and they ran the experiments, they saw that if you had been feeding on the leaves or the plants, then you have a very high reactive oxygen species generation in the protoplast when compared to the diet. So the response of the plant to produce ROS is much more in tomato plants if the caterpillar was feeding on the tomato plant. So clearly, there is a reactive oxygen species response, and there is follow-up with the lymph and other things that we are currently in the process of writing. So we were able to take it from the surface of the trichomes, then to the reactive oxygen species, which is the secondary messenger, and then there's calcium. And then, okay, so you are done with all of these things. You have eaten the plant. Now you have to process the toxins. So what happens with these toxins? So Dr. Somawat, who was a faculty here, he moved to Missouri. He had this interesting method of, Alex, how many minutes do I have? 5?
I mean, we're fine. I mean, it's up to you, really.
Right.
I mean, yeah.
I can go 10 more minutes then, yes. So this Dr. Somawat had found out a method to extract this pericarp from corn. And this pericarp in corn processing is actually a waste product. This is something that they dump. So he found a way. So there are ways to extract compounds from pericarp. He refined it, and his group has patented that. So, and there is this type of corn called purple corn, which is native to Peru, the Andes region. And this purple corn has a lot of polyphenols. And we know that polyphenols also act as secondary metabolites and are defensive compounds. So we asked the question, what if we get this extract and we add that into diet and see what happens when the caterpillars feed on them? Given that Manduka doesn't feed on corn, but we wanted to use Manduka as a test organism to see whether it will have an effect. So we had this yellow corn, which is just regular corn that we bought from Amazon. And then we have control, which didn't have any extracts. And then we had extracts from purple corn. And then we put caterpillars on them and we started to grow them. And then after a while, we would switch them. We would move from purple corn to control and control the purple corn and vice versa. And consistently, we found that if you are on yellow corn, then you do pretty well. Excuse me. If you are on control, you also do pretty well. But if you are on purple corn, you don't do so well. You try to compensate by eating more during the late stages, but the damage has been done in early stages. So we clearly show that it's the early stages of the caterpillar growth that it gets really affected by this toxin. So we found 2 things. One, this is a potential way of using this waste product, using this methodology. You just have to package it in a way and then feed it to the caterpillars. The second thing is the early damage, even if you can grow much more in late stages. So to put in perspective, the early stage caterpillars gain like 25 times their body mass in the first 6 days, and then in the next 6 days or 15 days, they gain like 600 times their body mass. So in that early stage itself, if you are exposed to toxins, then even if you move to a good diet, you cannot recover. And then Mandeep, so that was Mandeep's thesis. He also looked at the effects on other traits. So not just the caterpillars, he looked at the pupae, the pupal mass, their wingspan, and also fitness, realized fitness in the form of number of eggs laid. And then he looked at what is the mass of these eggs when they become caterpillars. So if any of your parents, so he did these matings, these pairwise matings, purple corn male, purple corn female, purple corn male, yellow female, and vice versa. Or yellow female and vice versa. So if any of your parents were on purple corn, then you are doomed, right? So the female won't lay a lot of eggs, but even if the eggs are laid, then they don't gain a lot of mass. So there is some cascading effects that actually move through generations and will cause continuous damage. So right now we are, so I'll say this, and so one of the questions that we had with our reviews were that, okay, so you have shown the proof of concept, but you need to show this with a caterpillar that would actually feed on corn, which was not our intention. But then we did that. We thought that was a good idea, and Supman did that. Supman is studying as a PhD student at Penn State, and Mandeep is now a PhD student at Clemson. Isabella is still in our lab. She got admitted to medical school. Annette is the person who draws all these nice illustrations for us. I forgot to acknowledge them. So Sukhman showed that with Spodoptera frugipeda fall armyworm, you can replicate the same results, which clearly shows that it is the toxins that affect, and there's nothing to do with the caterpillar or the species of the caterpillar. So we are trying to patent it, at least some part of it, and we are continuing to work on it. We are using nanoparticles now to see if we can package it better. The whole idea is to spray it and see if nanoparticles will help them to get stuck on the trichomes. So because these are water soluble, if rain comes, so things like that, and see how does the plants respond to that. So we have some collaborations with farmers to look at that. So now we have started from the trichomes. We looked at the early signal events of reactive oxygen species, and we looked at toxins, and we take them to the community. So this plant, the other plant that I work on is silverleaf nightshade. I started working on this when I was a postdoc. That's in Greece. It's highly invasive in McAllen in Texas. It's not that bad, but it's still a nuisance. Jesus Chavana, who was a master student in the lab, he started as an undergrad. He now works with Bayer, graduated last fall. So he started looking at following up my experiments from when I was doing in Greece. So he started looking at the data, collected data from the local region. Brad Christopherson is a colleague of mine, a good friend. He's a physiologist. So he helped us with some of these questions. So basically we wanted to ask, what happens if you are continuously mowed? So in McAllen and Edinburgh, the city mows these plants, and these plants come back very fast. So we asked the question whether mowing caused any effect in them at a multiple level. So we started looking at the fruit, seeds per fruit, and seed mass. What we found was, as we expected, because there is continuous mowing, they don't get a lot of opportunity to grow, so they produce less fruits and seeds. But the seed mass, so 100 seed mass, which is 1D here, is actually higher in a mowed fruits or mowed plants which produce fruits and seeds. And then we started looking at germinations. Again, a busy slide, but the gist is they germinated faster. So if you have fruits produced from plants which were mowed, when the seeds were planted, they germinated faster, they also germinated more. So clearly mowing is actually causing something in them at the embryo level, which is helping them to grow faster and grow more, or at least germinate them. And then we took them to the field, and then we put them out as seedlings and looked at herbivores. And we looked at 3 different herbivores. We looked at damage. We assessed herbivore presence. We also took them in the lab, and we put different herbivores on them. We looked at feeding damage and everything. And everything that we saw clearly showed that if you put a chewing herbivores and sucking herbivores, they vary, of course. And but in general, the mowed plants did extremely well. So this continuous mowing by the city is actually counterproductive, where if the parents are mowed, then the progeny do grow more, grow fast, get less damaged by the herbivores in the field, and they have less field damage, and the herbivores don't do well on them. Except for one herbivore, which is this Leptinotarsa texana, which is Texas potato beetle. It doesn't care, it doesn't matter. It's co-evolved with the species, so regardless whether you're mowed or unmowed, they do extremely well. So we are thinking that continuous mowing will make this plant a weed, a super weed. So this is what my lab toolkit is. So we have this push-pull based portable and fixed collection for volatiles. We have a GCMS with FID. We have an electrical penetration graph to look at electrophysiology of aphids and sucking insects. We have insect respirometry, so we can look at, we haven't used it much. Then we have Ethovision, which is a behavior tracking software. It basically tells you how they move, the vectors, what is their speed, what is their acceleration, direction, all these things. Then our most prized possession is this electron microscope, which you don't need any sample preparation. You can just plug and play, and you get imaging in like 5 minutes instead of like glutaraldehyde fixation or critical point at drying or weeks of this. And almost all my students are experts in all this. We also look at root architecture by looking at this WinRISO, that is a system where it will give you all the root area, fine roots, everything. We just bought a qPCR and an electroantennogram. We haven't got them yet. I'm sort of trained on electroantennogram, but not very good at it. It's the only company in Germany makes it, Syntex. So we just place the order. So hopefully we will get that. So from 2022, we can do some work with the bees, especially then the bee antennas. That's something that we are looking at. Like I said, the first part was from my PhD and postdoc. So Consuelo and Mark, whom I also did postdoc with at ETH Zurich, and Andy Stevenson. He just retired, distinguished professor of biology at Penn State. Then all my collaborators here. I didn't talk about USDA work, USDA ARS work on ticks with John Goolsby, but all these other collaborators whom I work with, these funding agencies, we had been lucky so far to get funding, and almost all the credit goes to them. So this is my lab. Some of them are students who already left. They came back for graduation, and we had a party. And this is the picture from that party. And with that, I will stop. And I know that it's 12:56. So if you have some questions, I can try to answer in like 5 minutes. If not, please find a way to contact me. Thank you very much.
Thanks to you, Rupesh. Yeah, fantastic talk. For those of you, it's a shame that Rupesh is going to leave like in 10 minutes, right? Rupesh?
Yes, yes.
Yeah, so if you have some brief questions, or if you don't, but in case you have brief questions, you can do that now, or get in touch with Rupesh directly. He'll be happy to answer that. And I was gonna actually answer something. Have you ever thought about looking at the cost of that injuries that the insects have after eating these trichomes, well, these leaves with these trichomes, directly from the insect perspective? Like, I was wondering, like, I don't know, with these injuries, I mean, the chances are the pathogens can have access to the insect and then produce an infection. Have you thought about that?
So Alex, excellent question. So there are 2 points. One is like, we can think from like an energy budget, right? So how much energy do they have to extra spend because they have to deal with these trichomes? And the second thing is post-ingestive effects, right? Because when you have this tear inside the gut, then it is an easy way for microbes to enter. And the follow-up of that question is, these trichomes, they are also considered as dirty needles. So they actually have some microbes at their tip, right? Like with spines when they would damage mammals, right? So they have these pathogens which would get in. So there is this group at Penn State, Dawn Luthi, and her student in 2002, his name was Pican. I think they are the first people who actually started looking at the pathogens. So there are many proteinase inhibitors in plants can cause this, and they have shown that it will help the pathogens to establish inside. So it has been done to some extent. Yeah, but more specifically, if you ask me, like, have you done that with Manduka? Because especially as a lab animal, no, we have not done. We are not microbiologists, and we have very little knowledge on these things. So we have not. So that's the answer.
Yes, well, anyway, I was saying that because we used to do lots of immunological measurements in insects, and actually also challenges using real pathogens. So we may find a way to collaborate on that.
Yeah.
Eventually, yes. Anyone else? A question for Rupesh? ¿Alguien que quiera preguntarle a Rupesh? Oh, here you have something. Laura, she's asking, well, she says, thanks, great talk. Can the differences in defense and volatile expression between inbred and outbred plants be related to plant growth?
Excellent question. And so when we did microarray, so there are 2 parts to the question. So let me see if I can explain. So when we did microarray, so we did a whole transcriptome. This was when microarray was hot, so like some time ago. We found that in inbred plants, the the growth related and housekeeping genes are, they have a higher expression, relatively higher expression. So those genes, their growth is actually better. So in field, you don't really see the inbred plants, they are shorter, or inbred plants, they are having less leaves, or you don't see that. But when it comes to fitness, when you look at the fruits and seeds, you see that effect. So if you look at very functional traits of growth, you don't really see that effect very much as far as growth is concerned. If that answers that question. The second question is, how can we explain the maintenance of inbreeding in plant population considering its detrimental effects, right? This is a very good question. So the answer is, with all these detrimental effects, you are still not at a complete fitness loss. You still produce fruits, you still produce seeds, the numbers are small, and we should always think about inbreeding as a way of sustaining local founding populations, right? So because you have very less number of alleles when you start with, because you probably thrown out into a new habitat. So inbreeding helps in that cases when these plants can just survive and produce a few fruits or seeds. The second part, in this plant, it has two more interesting things. It is self-incompatible. So it actually cannot inbreed unless there is no foreign pollen. So if you don't get an outbred pollen, then the self-incompatibility system will break down. It's an RNase-based system. So the younger flowers would wait for outbred pollen, and if they don't get it, then the next set of flowers, the older flowers will wait, and then the younger flowers will switch from self-incompatibility to self-compatibility. And making things worse, there's one more thing. They can also spread by roots. So a mother plant can actually produce new offsprings while the mother dies, and the next year you have new rhizomes which produce the shoots. So we did some genetic markers, and then we found that the outbred plants on average produce 21 new plants, whereas inbred plants produce 7 every year. So you have to think this, like in a founding population, yes, inbreeding has so much detrimental effects, but still there is some measure of fitness that makes them survive this onslaught by herbivores.
Mm-hmm. Yes, thank you. I think that answers the second question that she posed.
Yeah.
So, well, she thanks you, and also Idale Cooper thanks you, and some other guys. And I think if there are no more questions, I think it's time for us to leave this, because Rupesh is going to be teaching in a few minutes, right?
Yeah, I have to teach undergrads ecology in like 10 minutes, so yeah. I would like to leave soon. Yes.
Fantastic presentation, Rupesh.
Oh,
very nice. It was really, really astonishing, and very clever experiments as well. It's nice to see something so where everything sort of somehow fits with each other well, and altogether looks like a very nice piece of research. Thank you very much. We'll be in touch, Rupesh, and thanks to everyone for being here.
Thank you, Alex. Thank you, everyone. I'm going to leave then. Bye.
Yeah. Muchas gracias a todos. Que estén muy bien y nos estaremos viendo en 15 días. Muy amables. Ahí les queda el correo electrónico de Rupesh si quieren contactarlo. Y pues bueno, muy amables, y estaremos en contacto próximamente. Gracias y hasta luego.